Hi, today we're talking about the kidney and the nephrons within. All right. So the kidney is pretty complicated, so I'm just going to jump into it and we'll try not to take too long, but this is a big one, all right? So right now, I'm going to focus on the nephron. I sort of untangled it here, pulled it out like a garden hose, but the nephron is all coiled up and it looks a lot more complicated than this. Take my word for it. And so here we got our glomerulus capsule here, whatever. I should draw in a bunch of blood vessels right here. But we'll talk about filtration and all of that later. Right now, we're going to focus on how your body reabsorbs stuff, right? And so here is the stuff that's currently going to go to the waste if it's not reabsorbed. But never fear, the kidney is here and it has a plan. So first, we're going to look at the proximal convoluted, aptly named tubule, right? And so there's a little acronym I was taught that is really useful for this. It's called GOBWAC. And it's all about this, well, there should be a C there, but just take in good faith there is. And so this describes pretty well on what the proximal convoluted tubule does, because it does a lot, if not most of the heavy lifting. 70% of the water gets drained out of here. A ton of the solutes, your glucose, your amino acids, your lactates, like all that important gnarly stuff is getting out there, right? So just before I jump right in, here's the stuff that's going to get drained out here, right? Here's your tubules, whose job it is to reabsorb stuff. And surrounding those tubules will be blood vessels. I only drew it for this half of it, but it's like this all over the nephron. And so the tubules will be trying to pull the important stuff out. So the capillaries, via the process of just diffusion, can pick up those important chemicals and energy sources like glucose, for example, so we can keep reusing stuff. Right? Like this is called a reabsorption for the reason. Your tubule's goal is just to pull everything important out and leave all that crap behind so it can get peed out, right? And we'll talk about pathologies and the like later, but we're jumping right in here, right? So first we're going to start with our amino acids, your glucose, and your lactates. They function the same way. The sodium inside of the waste gets drawn towards the tubule and bringing with it the important stuff we all love so much at 100% efficiency, which is fetching crazy. So that's one of the reasons why our body can use glucose so efficiently, right? It can just 100% reabsorb all the stuff it didn't use the first time. And you might be asking, Nick, why does sodium even want to get here in the first place? Well, you see, there's our beloved sodium-potassium pump, right? And my apologies, my foot just kicked it. And the sodium-potassium pump pumps out all the sodium, causing a drive towards the tubule. And so it'll artificially create that gradient. You know, we love artificial gradients here in physiology, right? It's literally our bread and butter. And here, this is also important to mention because, you know, it's how CO2, one of the ways CO2 can get produced, right? And so protons will go into the waste, right? It'll interact with the bicarbonate there, and via the anhydrase there, produce CO2, which is super easy to diffuse, and then it can get out and go through, and then back into our blood vessels, where we can breathe it out with our lungs, right? And so, what else am I missing? There'll be channels for all this stuff here, like the special channels we should pay attention to here, such as GLUT2, which is your glucose channel that enables it to get all the way through. Your SLGT, which is what I think it's the pump that the glucose uses to get in the tubule in the first place. And there'll be channels for all this stuff too, right? To get it out of the tubule. And so spend a lot of time to study this at home. It's complicated. Sometimes I still need help getting my head around it. But another important thing to know is the water drag, right? And so water will be leaving via these junctions in between the tubule cells and all of that. and with it, it'll bring calcium and potassium, which is your whack. Water and calcium, there should be a C there, sorry, and potassium, right? So glucose, amino acids, then the L is lactate, then the B is blood. So to the blood is glucose, amino acids, and lactates. Then water drags potassium and calcium. And so this tubule actually pulls most of the water, right? And because of that, you would think it would drop or increase or do something to the osmolarity. But the thing is, so much is happening at the same time, like pulling out all the glucose and other particles, that it actually just stays at a constant 300. Now we have our my apologies, I misspoke there, our collecting straight tubule, better than those bendy tubules, right? And so we have a lot going on here, right? We're looking at our waste beaker here. We have our sodium getting pumped in, protons getting pumped out, protons getting in, anions getting out, chloride coming in, anions coming out, and those will interact with the protons, right? Because they're negative, so they'll solve each other. We have our beloved sodium and potassium. We're never escaping it. Like, it's just going to be there forever. Frick. Well, and it's all right. It's not too bad. And then you have your chloride channels. These are your cystic fibrosis channels, right? They're super important. If they don't work, you're cooked. You get cystic fibrosis, and not to make light of it, it's a serious issue, tragic. But you're probably dead before the age of... No, you will be dead, most likely by the age of 25. If these channels don't work, because your mucus won't work properly in your lungs, right? And if your lungs don't work, then yeah, it's not too fun. It sucks. There's aquaporin channels, so water can go through there, and like I mentioned before, these junctions in between, right? And the osmolarity stays the same. Now we have our descending tubule here, and it's just water. Right, you have your aquaporins, flies out. You have the holes between the cells, flies out. This is very water heavy, but don't let that deceive you, because this is still your Mr. Big Shot, your proximal convoluted tubule, and that's where the majority of your water heads out, even though you would think the one that's primarily water would serve that purpose. But hey, the world's a strange place. I think that's about it. Something I like to use is descending tubule as a benchmarker. It's a really good benchmarker, because after this, you would think there would be there's no more holes between the cells. There's no more those open junctions between the cells. So it'll be buttoned up, and no cells or no water will leave in between the cells. If water leaves, it's because there's aquaporins. So moving on to the ascending tubule. Cystic fibrosis channels back on the table, back on top, big dog. We have our sodium 2-chloride potassium channel, pretty unique, pretty cool. I think it's nice. You might not, but it's pretty rad, to put it colloquially. Back in black, we have our sodium potassium pump. Frick, I love it. I hope you do, because it's not going anywhere. By the way, the descending channel had it too. And then you had a potassium channel, and then like I mentioned before, your cystic fibrosis channel. Again, it's important to have these. If you don't have it, dead as a doornail, unfortunately. And so you'll notice it gets super concentrated down here. You can't really see it because my computer is lame and small, but that's okay. Down here, it's 600 osmolarity, so very concentrated. Up here, it's much less concentrated. And here we have our macula densa. And our macula densa is quite immaculate at doing its job. So it'll draw bicarbonate from the outside. Wait, I think I might have drawn this poorly. I might have made a mistake here. My apologies. Let me just check something real quick. Well, sodium comes in, right? okay, what you really need to know about the maculodensa here is that it's used as a sort of detection to see, like, okay, how much sodium are we looking at here? And depending on that, it will help work with a globotubular response and also indicate the import of ADH and aldosterone stuff I'll get to later. But that's really what we need to know right now. And assuming my diagram is correct, it'll draw sodium and okay, sorry, I was just turned around for a second. It'll draw out sodium and chloride from the waste, and then it will keep pulling out chloride into the outside so capillaries can drink it in, right? Because we are all about that chloride. It's really important. And then bring back bicarbonate. And there's some reactions we can do here to help out in this process of reabsorption and even filtration using the maculodensa mechanism, but we're just going to keep moving on. Now we have our distal collecting tubules here, our principal here, right? And so we're here with our waste. There's channels for the potassium, which brings it out, sodium brings it in, and then we got our sodium and chloride pump. That's pretty neat. We're mixing things up now. And potassium can also get drawn out via that way too. It's just your body's working really hard to just get everything it can out of the waste so it can reuse, right? And it does a pretty fetching good job. And so now we have what I like to call the A section or the acidosis section of the distal collecting tube. And so you'll have this G protein which can detect ADH, and ADH will create aquaporins. What purpose does that serve? I'll get to that, but you just need to know that for the part A of the distal collecting tube, the type A cells, is that it'll pump out protons in order to reduce the acidity of the blood, right? It'll pump, sorry, I had to sneeze for a second. And then it'll pump potassium into the capillaries and draw out protons. And then B, which is for alkalosis, the type B cells, if there's too much, what they will do is they will pump out protons and pump in potassium, and then also sort of pump out bicarbonate too, but this is its primary function, I would argue. Oh my gosh, I keep having these half sneezes. It's fetching atrocious. Okay, so I totally speed ran through that. But that took me 13 minutes, and we're about a third of the way through. So we're going to keep gunning through this. And this was the process for absorption, not filtration. We'll get to filtration in a sec. But before we move on, I'm going to cover the idea of renal reabsorption and all of that, and your limits and stuff you should know. So I'm going to use a common example for diabetes, right? For diabetes, there's an important channel right here that I mentioned earlier in your proximal convoluted tubule called GLUT2 or whatever. And GLUT2 is good as it jobs. It chauffeurs the glucose into the blood, or plasma, I should say, which can be absorbed by the capillaries, so it can reuse that energy. But the problem is no matter how efficient you are with your body is with your glucose intake, if you drink like 20 sodas a day, brother, you're going to have diabetes, right? Because these have a limit at which these proteins can carry the glucose into the blood. And if you overload that limit, which is pretty fetching hard to do, but if you somehow do it, then the glucose will just slip on through into the waste and out for excretion. And then you'll have glucose in the blood, and that'll tell your body, or your doctor, I should say, your body won't know what's going on. It's just a machine. But your doctor will look at it and be like, shoot, you have diabetes. Now, whether that's naturally induced or insulin or yada yada, that's for your doctor to decide. But that is an example of your threshold and limit, right? Because you have your glucose threshold, you surpass it, and your body will fly right through it. And you saturate your channels, and that's what happens. Okay, so I talked for 15 minutes on that subject, but that was really important. I might even revisit it later, but we're going to keep moving on using my fancy technology of trash can on a table. I'm going to magically move this computer with perfect stability. I could have been a cameraman in a different life. All right. Let's see if that's good. Perfect. Now we're going to talk about filtration, and this process starts before reabsorption. All right. I'm going to cover... Oh, what do I want to cover first? We'll just go through this, right? So you have your afferent capillary. Should have written this out beforehand, my apologies. And efferent. And so your afferent capillary will bring blood into this web of capillaries right here, right above the little glomerulus capsule. Sorry, I struggle saying that word. And then it'll run through the fenestrated endothelium found within the capillary, right? It'll go through the podocytes between this gap here. And then it will go through the basement membrane, right? And that's only if the stuff can pass through it, right? To help filter what should get excreted and what shouldn't, right? So let me give you an example of stuff that can't or can be filtered, right? Blood is way too big to go through the fenestrated endothelium found within the capillaries. Blood is like 5,000 nanometers long. The holes within the fenestrated endothelium is 50, right? So we know if something needs to go through this filter, it has to be really small. It also can't be charged, or especially negatively charged. So large proteins in blood, stuff like that, will bounce off and not get filtered through. Smaller stuff like inulin, which is, I will be returning to the concept of inulin later, or just really small particles like chlorine or sodium, they can slide right through the podocytes and through the basement membrane of the glomerulus or whatever and get right through. And now you're probably thinking, like, all right, this is cool, but how does this actually happen? Why would this stuff even want to leave in the first place? Well, you see, we're returning to the idea of pressure, right? Pressure, our beloved, right? We didn't have enough fun with it in a cardiac part of this class, so we got to go back to it. We get to go back to it. And so we have, I'm going to throw some numbers up here, and then I'm going to reiterate what's really important, the message that's trying to be sent. So you'll have a hydrostatic pressure of 55 in the capillary. Then you'll have a B cup of 28, or blood colloid osmotic pressure, which is pretty much just the pressure drawing into the capillaries. And then you'll have the hydrostatic pressure of the glomerulus itself at 15, right? So let's do some math here. You'll have 15 pushing back into the capillary, 28 pushing back into the capillary. We'll just call it at an even 45. And then you have a total of 55 hydrostatic pressure pushing out. Right, 55 pressure pushing out. And so you'll have a pressure of about 45 wanting to go in, but 55 pressure wanting to go out. And that will lead to about 10 total pressure pushing out of the capillaries, enabling this filtration process. So just to run through it, because I think it's interesting. After filtration, you'll have the reabsorption, which we already talked about before, right? Back into the capillaries surrounding the nephrons. Then your nephrons can also secrete stuff. We didn't talk about that as much, but there were still examples of stuff going back into the waste, right? They'll secrete it back, and then it can excrete that stuff later. So for example, for the type A cells in the distal collecting tubule, there was examples of protons getting secreted or pushed back into the waste of the body. And that means, like, when you're suffering from acidosis, you can, now I'm theory crafting here, I'm not a certified doctor or nephrologist, but that would lead me to believe you can almost determine acidosis from a urine sample, Because if you have an excessive number of protons within your urine, then your body's working overtime to fight that acidosis. And all right, perfect. Now we're going to revisit an old friend called macula densa and what it does to help us. I briefly went over earlier. Remember, it was after the ascending tubule, but before the distal collecting tubule. And I talked about how it can release certain chemicals if the pressure isn't high enough in the blood, or the sodium chloride levels aren't high enough in the blood, which is a good measure of pressure as well. Sort of a happy accident sort of deal. I shouldn't say accident, but sort of a beneficial occurrence for us. Because if your macula densa detects that I'm going to bring this back over here because I just want us to look at it here. If your macula detects that the pressure is too low or too low sodium, what it can do is it can release nitric oxide, which dilates the afferent capillary, which increases what is called GFR, which is sort of like the glomerular fluid pressure or whatever, which is like how much pressure is running through it right now. And then if it's too high, then your macula densa can release adenosine, which vasoconstricts your efferent capillary, which would also increase it? Or would it vasodilate it? Regardless, your body can release certain chemicals it would vasoconstrict efferent, that shrinks it. Oh, I might have confused that. Maybe adenosine vasodilates. Regardless, what you really need to know is that your macula densa can release chemicals in order to adjust the GFR. What's the GFR? I'm glad you asked. Even though I already sort of answered that question a bit. Here, sorry, I messed this table up. Perfect. All right. So GFR, I don't know what this is doing up here. It's sort of like your glomerular fluid pressure, right? And it's heavily correlated with this right here, your clearance rate. And your clearance rate is how much solute is dropped from your body in ratio to the plasma that is dropped from your body. It's pretty much how effective something can be reabsorbed, secreted, then excreted, right? And so the GFR will actually equal the clearance rate here if you eat something or inject something into yourself that's small, not charged, and can easily slip through. Sorry, I'm trying to do this while looking forward like a weatherman, and it is rough. And easily pass through the filtration, right? We want something small that just flies right through to excretion in order to detect how efficient your GFR is, your fluid pressure, right? And so you can pee it out. And so scientists used inulin. That's a long story, but it's just a small non-charged particle. And we can use it to detect GFR. And so we also learned that there's a chemical reaction when GFR is low. And this is other stuff that I think is super cool. If GFR is low, then renin goes up. More renin, that renin turns into angiotenogen. Angiotenogen turns into angiotensin 2 via ACE. After angiotensin 2, you'll remember this from the cardiac stuff, right? Remember when blood MAP or mean arterial pressure was low, then your body would release this as well, renin, or your kidneys would. We're running it back to that. So if you remember that, then it's very similar. But now we have these two fun additional functions. There's a lot of fun in this conjunction here, right? Because it will release ADH, which increases the aquaporins found in your distal collecting tubule, and it will increase your aldosterone, which increases the number of sodium potassium channels, right? And so this has the really neat effect of, well, think about it. If you increase the number of aquaporins, then it'll be easier for water to leave the excretion pipe or whatever and back into the blood, increasing the efficiency and reabsorption of your body that way. And by increasing sodium potassium pumps, you make your urine more, what's the word I'm looking for? Concentrated. That's the word I'm looking for. Yeah. And so I believe that's it. I'm going to check something in case I missed anything. Yeah, your adrenal gland is in charge of aldosterone. That's good to keep in mind. Okay, let me just check to make sure I didn't miss anything. I think that's about it. Yeah. Clearance rate, we covered that. Yeah, this whole GFR thing, your macula densa helps work with that, because if GFR drops, then it can artificially increase it by either dilating the afferent, by either, it's opposite day. When you dilate stuff in the kidney here, it actually decreases mean arterial pressure, not increase it. So if you dilate the afferent capillary, then it'll actually increase the map here. And if you vasoconstrict the efferent, that's what adenosine does. It vasoconstricts the efferent here. Wait, does it? Oh, that's so annoying. I can't remember exactly. Or you can vasoconstrict the efferent, and that would also increase the mean arterial pressure within the glomerulus here, and that would increase your GFR, helping to restore balance. But these are just two additional toolkits that your body can use to help deal with that. Yeah, no, I think that's about it. Thank you for watching. Have a marvelous day, and thank you.